Hi class, Phil Sack here. In this video, I would like to talk about barcoding, DNA barcoding. So DNA barcoding is a method that is used to basically match DNA sequences to, or genetic sequences to a database. And the purpose of that could be, for example, that you want to retrieve the species based on a DNA sample, for example, okay? Or you want to retrieve maybe a protein sequence or a sequence of RNA and so on, okay? So just a quick reminder of what is genetic material and what do we need to before we start actually tackling the problem of matching sequences, okay? How do we get these sequences? So we get this first. So as you probably know, genetic material is mainly composed of nucleotides. And these nucleotides come in several flavors, and that I've shown the sugar part of the nucleotides here. And I have like these three diagrams here, A, B, and C. And I'm showing here three types of nucleotides, nucleotidic acids. One is ribo, which has two. So the carbons are, this is the sugar part. So the carbon position are typically labeled in this order, 1, 2, 3, 4, 5. Okay. And you can see that there are some hydroxyl groups at position 3 and 4. Now, depending on the types of depending on the presence of hydroxyl group or no hydroxyl group at position 3 and 4, you will have a different type of nucleotide. So you have a ribonucleotide if you have OH and OH, so two hydroxyl sites. Okay. You have a DNTP if you have, or deoxyribonucleotide if you have OH at 4, but only H at 3. And finally, we have D-deoxyribonucleotide if you have 2H. So, okay, neither do you have a hydroxyl group at 4 or 3. Okay, so now what we have to mention is this. In 1977, Frederick Sanger realized that he could take benefit of this one here, this species, this chemical species, this type of nucleotide, to stop the polymerase, DNA polymerase reaction. It was a very smart idea. So as you probably know, if we take you know that DNA is a molecule that has two strands, okay, of atoms. And these two strands are homologous, and they are basically, it's a double helix, okay? So hard for me to write this, but okay. So you have two helices, two helices, okay? And they are bonded by hydrogen bonds, okay? Hydrogen force. Weak forces, weak atomic forces. Okay, and if on one side you have a base that is A, for example, you have the conjugate base on the other side. Okay, you have, for DNA, you have 4 bases A, C, G, T. Okay. So at A, T and A are conjugate, and C and G are conjugate. So A goes with T, C goes with J, J goes with C, and T goes with A, like this. Okay. All right. So if on one strand you have those here in this order, on the other strand, you have the other guys, these other guys. Okay. Okay. All right. So what happened when you want to sequence the DNA of some organism is you can use the classical method of Sanger. And as I said, Sanger took advantage of the fact that Taq polymerase stops if it meets the Taq polymerase, okay, induces polymerization of the reaction. So we have the template here, one strand, okay. Here you have your primer, okay, and then you put in solution some nucleotides, and they get added here, okay. Except that to add them, you need to have this radical here, okay? If you don't have it, the reaction stops, okay? So if you keep adding, okay, the reaction keeps adding bases here, okay, that are conjugate to at each position to the other side, okay, until it hits a DDNTP. So the idea of Sanger, okay, was to actually use 4 tubes. In each tube, you put a primer, you put the nucleotides, you put a template, and you put radioactively labeled DDNTP. Okay. So you put DD-ATP here. So you put okay, this here the radiolabeled sequence carry a radioactive A is a DDNTP here terminates with A or carries A, okay, and so on. Okay, here DDNTP carries C, DDNTP carries G and T. Okay. So what happens is that in the first tube, okay, you only generate segments. All the segments stop, the polymerization stop at A. So all the segments finish at A. In the tube 2, all segments finish at C. Tube 3, all segments finish at G. And tube 4, all segments finish at T. Okay? And then what you do is you just put your segments, or you produce the products of your reaction onto polyacrylate gel, and you do an electrophoresis. And the heaviest fragments, they migrate less far, and the lightest migrate further, okay, because they are their mass is less, okay. And so you end up with after exposing the gel to X-rays, you end up with what is called an autoradiogram. And on the autoradiogram, you can basically read the sequence. Okay, so here we have on this axis here we have basically we have the base number, okay, position in base number. So number 1, number 2, number 3, and so on. Okay, and you can just read out the sequence of your sample. Okay, so that's basically the principle of the method of Sanger for sequencing DNA. Okay, and you can apply this idea for RNA and so on. Okay, so another remark is that we don't have to use 4 tubes. If you can label each product, like each base, A, C, G, T, if you can label them with different color, for example, with fluorescent dyes, okay, well, then you can just use a single tube and go much faster, and put everything in one tube, and with a fluorescent dye, you basically get your sequence as a series of colors along a line, basically. Okay. Okay, so now step 2, I said that I would talk about DNA barcoding. So how do we use these sequencing methods? And today, well, we would talk about probably about next generation sequencing. Well, that was the original term. Now, next generation sequencing. Probably there has been several generations of next generation sequencing. Okay. And well, I'm not a specialist of those, but they are basically ultra fast and efficient ways to sequence genetic material. Okay. Okay. Now I come to step 2. So DNA barcoding. So what do we use that for? So now that we have our sequence, it's a way to sequence DNA or RNA or proteins. What could we want to do? Well, there are many applications. For example, we may want to recognize species from the DNA sample or recognize a protein sequence. And the idea is very simple. We would just align, align our query sequence. So we call this a query sequence. Here, query sequence. So that's the sequence that we are interested in, okay? And we want to gather information about this sequence. So basically, we are simply going to compare this sequence with sequences or one sequence or several sequences that are labeled or annotated, and then they are in a dictionary or in a large database, okay? So here is basically the sequence I have. I have I've analyzed, okay, and now I want to match it maybe to a number of sequences with their labels, okay. And so you can do that online, and there are many tools that allows you to do that. This is very popular exercise in bioinformatics. Tons of sites do that, okay. Well, the simplest example of this exercise would be when you want to match, say, like a short sequence here, with you want to retrieve the sequence or match it, align it somewhere in a genome, okay, or in a very long sequence contained in a database. Maybe you have like genomes of various organisms, and you want to see if that sequence here belongs to one organism rather than to another, okay. So we have to, when we do this kind of matching exercise, we have to develop similarity scores, okay, between sequences. So that we are going to manipulate sequences of letters. For DNA, we'll have 4 letters, ACGT. For RNA, we may have 20 sequences, and for codons, we may have 60 letters, okay? So the size of the alphabet that you are using will depend pretty much on what you are doing, okay? And so there's basically a range of variants, basically. Okay, here the simplest variants are ACGT. Actually, what the method that that I will be talking about are totally general, and they would work for any kind of sequence. Okay, sequence of letters or sequence of symbols, as long as you have some variation in the population and you're interested in somehow in a sequence of symbols. Here, genetic material. Okay, so similarity scores, we can look at them as metrics or like distances. So we take one sequence, another sequence, and we measure essentially either a distance or a similarity between the two. Okay. Now we can encode this. For example, if you have R letters, okay, for example, 20 amino acids, okay, 20 amino acids. Well, R would be 20, and then the way to score, you basically would totally determine the scoring by a scoring matrix that would be 20 by 20. Basically, we would write Sij for the score of comparing AI to AJ. Okay, letter AI to letter AJ would receive score Sij. Okay. Yeah. So S is a common, well, it's actually the symbol that was used by the authors of BLAST. I will come back to that in the original paper. Okay. So now we realize that when we try to match sequences, okay, we are primarily interested in some form of scoring, okay. And the simplest form of score is, of course, the following one, okay? And I'm going to concentrate on that one in this simple video, because this video is not for experts. It's just a video to give you the central idea of the statistical methods or mathematical method behind the alignment tools. So the score, the simplest scoring method would be to say, I increase my score, my similarity score by +1 if, say, I match a letter here in the same position, okay, in my sample and my target organism sequence, or I have an A in both cases, okay, so that would be a match, so I would increase my score by 1. Similarly, I would increase my score by 1 if I had CC or GG or a TT. Okay. And for all other cases, for example, AC, when the letters do not match, okay, again, the same position, okay, I would just remove 1 from the score. Very simple. So basically, well, each time the letters are at the same position match, okay, increase by 1, in don't match, decrease by 1. Okay, I'm not here talking about insertion, deletions, mutations, and gap in sequences and so on. Okay, I keep things simple, and I'm going to use that very simple scoring method to illustrate the methodology. However, okay, the even if the model I'm going to talk about and to present is very simple, it is the essence of more general models. Okay, so it's always better to go first through a simple model, and then you easily understand that it can be generalized. And this is my approach. Okay, so basically, what are the problems now when we are talking about matching sequences? Well, we can think of 3 problems. First, we need to define correctly what objects we are trying to detect. Okay, that's 0. 1, well, we have probably to develop methods to efficiently find matches. Okay, so that's the area of dynamic programming and algorithms for basically very rapidly finding matches in huge databases. Okay, and this is a very important area in which bioinformaticians excel. Okay, and finally, and this is the one I'm going to look at here, we have to see how or develop a method to decide if matches are real or statistically significant, or they just occur by chance. Okay, so this is critical. Okay, because this is critical unless you understand this, well, you don't really understand what's going on. Okay, so that's basically, I would say, the key to okay, except that for the idea of sequencing, I would say that this is really important. Of course, every step is important, but if you have no way to decide, where are the matches, and what do they, I mean, are they significant or not, well, then basically you can't progress. So of course, people have developed methods to address these problems, and the main software, the most popular software, is called BLAST, which the letters of BLAST stand for Basic Local Alignment Search Tool. Okay, so it's really a tool, and this tool actually earned the paper in which this tool was published, first BLAST paper, earned no less than 100,000 citations. So it's one of the probably most cited papers on the internet. Okay. This work was done in the 1990s. Okay. And okay, the idea was that this tool was actually designed to find local or short sequences with very good level of matches, and that was a kind of move away from a previous approach in which one was focusing more on global alignment. Um, so it was realized that the protein domains were discovered. So it was discovered that short sequences can be very significant. Okay, you don't have to, you don't have to actually always look at the entire sequence. If you can find very relevant pieces of information by matching, subsequences, basically. Okay, so BLAST was designed precisely to maybe find maybe a collection of subsequences that match that match your proposal sequence. Okay, and the mathematical analysis, the statistical analysis, was mostly achieved by Sam Carlin, who is a renowned statistical geneticist. So the BLAST paper was written by SF Altschul, Gish, Miller, Myers, and Lippmann. Lippmann was the supervisor of Altschul. And then, in parallel or together with some of these authors, in particular Altschul and Carlin, there was development of the mathematical analysis. Okay, so put the mathematical analysis on firm grounds. And the analysis of Carlin, Dembo, and so on, actually are a substantial part of BLAST. Okay, and basically the fact that you could compute p-values and test hypotheses in BLAST was a key selling point for BLAST. Interestingly, the mathematical method, the main item in the basic theorem, if you want, that the mathematical analysis of matching of sequences is based on, basically it's a result that was proven many years ago in 1972 by Eigelhardt, who was a student of Sam Carlin, and actually Eigelhardt won the John von Neumann Theory Prize for his work on waiting queues. Here is his 1972 paper, here. And that's this paper that contains the key to the analysis made by Carlin. Okay. Now what I want to do is step 3. I want to actually show you, okay, a simple model that will make you understand how we can develop a quantitative method for alignment. So the idea is this. I'm going to consider a template sequence here. So, template. I'm going to say that my database is simply my database is made of one long sequence here. Okay. And here I have my query sequence. Okay, so my query sequence could be G, G, A, G, whatever. Okay. Okay. Actually, let me maybe pull some file here. Just okay. So yeah, we could look at you can look at this sequence. So okay, so here the top one would be the database sequence, and the bottom one would be one sequence that you have to and you want to query. Okay, so what you want to do is you want to analyze the similarity. Okay, and align basically the bottom sequence with the top sequence. Okay. So align. I mean, if we have to give a sense to the word align, it means basically that we want to maybe position the subsequence, the sequence below, okay, at the right place under the top one, and find basically common subsequences. Okay, relatively, long common subsequences. Okay. All right. So so what we can do, okay, is we can go, okay, what we are going to do is we are going to calculate a cumulative score. Okay. But I want to first show you my null model, okay, because we are going to need to compare the match we find to chance. Okay, so we want a random model of sequence, and our model will be simple. Okay, we are going to assume, okay, that we have an alphabet of letters. Here we have actually 3 letters, so this is A, C, G, T here. Okay, And we are going to assume, okay, that each letter has its probability. And in the chance model, okay, we are going to assume that these letters occur basically randomly and independently. So basically, if we observe a sequence of length n, okay, and we are going to assume that each letter is identically independently distributed from this probability distribution. Okay, and what we want to do is we want to analyze the cumulative similarity score. Okay, so let me show you how we can actually. So because the letters are basically random. Okay. Okay. I will have a probability of P, okay, of match, and a probability of Q, let's say, of non-match. Okay, so this is going to be our very simple model. Okay, I'm actually not going to really analyze what happens in detail to A, C, G, T. No, I'm just interested in actually a match or non-match. Okay, and again, remember, a match, it means that I increase my score by +1. Non-match, I decrease my score by 1. Okay, so this way, okay, when I actually do my comparison, here's time. Okay, when I progress with my comparison, what I can do, what I can see, I can picture what's happening here as basically a random walk. Okay, so you can see here, sorry, because this is actually capturing automatically. Okay, so what I'm showing here, this is a score. Okay, and this is time or position. Okay, and this is a cumulative score. So you can see that what I'm doing here is if you follow what's happening here, okay, my score increases first, then decreases, okay, then increases again, okay, then decreases again. So I'm doing on this axis here, I'm doing a random walk here. Okay, I'm doing a random walk. Okay, so if I write this y-axis or s-axis, okay, if I write that like here, okay, horizontally, okay, basically you can see that my score fluctuates. Okay, and I'm doing by I am actually I'm doing a random walk. Okay, and sometimes I can have large excursions. Okay, that's what's happening. What's occurring. Okay, so now let me bring up another image. Okay, to show you, to show you an example of that. Okay, so what's going on here is this. Okay, if you look at these, if you look at the the points that are like these points, okay, field points, they are called ladder points. Ladder points, and basically they are the absolute minima so far. Ladder points, okay, equal minima, absolute minima so far. Okay, so each time I beat my previous absolute minimum, I create a new ladder point. So you can see here, the first ladder point is there. The second ladder point is there. Okay, I'm trying to align this like this. Okay, third ladder point is there, and so on. And ladder point can only decrease, of course. Okay, because I need to beat my previous minimum. Okay, for example, here in this sequence of ladder points, okay, I decrease badly my similarity. Okay, and in between ladder points, you have excursions with highs. So basically, you can picture the random walk, okay, which accumulates a score as something like this, okay. Here you have your ladder point. Okay, your starting point. It's a ladder point. Okay, you do something, fluctuate. Okay, and then you beat your ladder points. Okay, you get more negative. Okay, so again here from this point here, I need to go up. Of course, otherwise it would not be a ladder point. Or yeah, and actually no, you can also go down and create also. So I'm going to go down one more. Maybe go down. Okay, and now okay again, and so on. Okay. So these are the ladder points here, 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 here. And what we are interested in is the excursion of highs. So here's a high, y1. So y1 is the maximum between after the ladder point and before the second one. Y2 here, okay. Y3 here, and so on. Okay. So now what we want, okay, question. What is the distribution of these y's, these yi's? Yes, A and B. Well, what is the distribution of the maximum of these yi's? Okay, so that would mean, what's the distribution of the most extreme matches, highs in match? Okay, you can see that the y's are fluctuating. Okay, and we want to know how to represent that mathematically. Okay. So how can we answer these questions? So what we are going to do is we're going to do something very simple. We are going actually to place two walls, one here at S equal B, and one there at S equal A. And we are going to start somewhere here at H, okay? And we are going to do our excursion, okay? Random excursion. And we are going to stop when we arrive at one of these barriers. We call this absorption or capture. Okay, so we have a random stopping time, and we either are absorbed by B, by this, or we are absorbed by A. Okay, either one or the other. And now what I'm going to do is I'm going to, let's say use the notation WH. I'm going to call WH the probability that we are captured by B. We are captured by B. Probability that we are captured by B, given that we started at H. And we are going to call UH the probability that we are captured by A, given that we started at H. Okay? The question is, how do we calculate WH and UH? Okay? And also, what the kind of question that we are interested also in is, how long will it take, on average, how long will it take, on average, before I am captured by B? So when I say I am captured by B, okay, this suppose, of course, that I'm not captured by A before. Similarly, if I'm captured by A, okay, well, the game stops at A, okay? It's a random stopping time, okay? And and we want to calculate these probabilities, okay? So what parameters do we have? It's surprisingly simple model, very simple, because if you think about it, how many parameters do we have? In fact, we have one parameter. Just amazing, right? Single parameter. Do you know which one it is? In our entire model, how many parameters do we have? Well, if you think about it, we have only one parameter, which is P, the probability of a match. So what I'm going to do is I'm going to assume, okay, well, of course, probability of P plus Q is 1, clearly, because match and non-match are complementary. Okay, I'm going to assume that Q is larger than P, okay? So P is smaller than Q. Probability of a match is smaller than probability of non-match, okay? And that's because I don't want to, well, basically I want to have subsequences and not to match the entire sequence, okay? So I want, I eventually, well, yeah, this is an assumption that is important. Okay, so the question is, how can we calculate WH? So let me pull out the method here. Okay. So to calculate WH, we are going to show that WH obeys a certain evolution equation, basically. So we are going to say, okay, that WH, okay, is the probability, just a sec, because my pen is difficult to use here. The probability of arriving at B captured by B at score B, okay, when I started at H, is certainly the same, and I'm going to use the law of total probability. So I'm going to decompose that H on the right and on the left. À la Kolmogorov. Okay, so if I read that, it's quite evident. The probability of B given H is the probability of B given H minus 1 times the probability of H minus 1 given H, plus the probability of B given H plus 1 times the probability of H plus 1 given H. So I'm starting either from H plus 1, so it's like this, okay? Instead of going from H to B here, I'm actually doing that in 2 steps, okay? This H plus 1 here, and that's H minus 1, okay? So of course, to transit from here to there, okay, it's a product of this probability here, and to transit from or or from here to there. Okay. Okay. So this formula is clearly right, and so this, by definition, is H minus 1. And the probability of losing 1 point is Q plus, and that would be WH plus 1 P. Okay, so I arrive at this recurrence relation. Okay, I arrive at this recurrence relation. Okay, now 1, do you agree that 1 is P plus Q? Therefore, I can say that WH is also P plus Q WH. So that's equal to P WH plus 1 plus Q WH minus 1. I've not changed anything, right? And now I can rewrite this easily as WH plus 1 minus WH divided by WH minus WH minus 1 equal Q divided by P. So this is a very important formula, and I can see now, okay, so I'm going to call this parameter rho. And so what I discover is that actually there is a geometric, there's a geometric series for everything, okay? So using this relation here, okay, using that relation here, I can easily calculate, for example, WH plus 1 minus WH. I can divide this by WH minus WH minus 1, multiply that again, okay, like this, and continue to decrease H, continue to decrease H, until I arrive at WA plus 2 minus WA plus 1 divided by WA plus 1 minus WA, okay? And so this simplifies, okay? And you find just WH plus 1 minus WH divided by WA plus 1 minus WA. And this is, of course, rho power H minus A. Okay, because we have H minus A products. H minus A, H minus A terms, and each term, each is rho. Okay? All right. But now, okay, I observe the following. I observe that my evolution equation here, this is my evolution equation here, evolution equation. I observe that it has some boundary conditions. Indeed, if I start from B, okay, I am sure that I will arrive in B. And if I start at A, I am captured by A, I don't continue, so I never arrive in B. So I need to, I can, and I need to use these two conditions. For example, here, WA here is 0, okay? So therefore, okay, if you put everything together, you easily, very easily, arrive at the following equation. You arrive at basically a full equation for WH. You can solve for WH, and the result is surprisingly simple. And again, this is probability of B given that you start at H. Okay? Similarly, you can write that UH is 1 minus rho H minus B, okay? 1 minus rho B minus A. And this is a probability of this. Okay? All right. A minus B here. Okay. So basically, I've been able to calculate everything. I've been able to calculate everything in closed form. Okay? All right. So now that we have that, okay, we can go further. So let me show you the rest. So okay, so suppose that we call n the random number, okay, that we need, of steps that we need to perform before we arrive at a score of B, okay? We are captured by B, okay? And now I'm going to call MH the expected value of n, well, given again, of course, that we start at H, okay? Well, we can easily show that MH minus 1 is P mh plus 1 plus Q mh minus 1. Now this is separate here. This is not M index, this is mh minus 1, okay? So if you want, I mean, if this is not clear, I can put it like this, okay? And for these boundary conditions here, now I have MA equal to MB equal to 0, of course. These are the boundary conditions, okay? So again, we can easily show that the following equation holds. Okay. So here I'm actually using here my calculations here to calculate MH. All right. So we have done a lot of progress because we can calculate a lot of things. We can actually calculate, you know, the time it as a number of steps it takes to, as you can see here, to reach a certain score. Okay, on average. Okay, this is an expected value. Expected value. Okay. Okay. Now we can also easily show the following. So now we are going to say, okay, we are going to take for A, we are going to take A equal to minus 1. Okay. And for B, we are not going to take any limit. Okay. We are going to take, actually, B can be as large as possible as we need. Okay. So in fact, for B, I'm going to take a value Y here. Okay. And I'm going to ask, what is the probability that my cumulative score is less than a certain value Y? Okay. Well, by applying what we have seen above, okay, this is given by rho power 0 minus, I'm going to start at 0, so I'm going to take h equals 0, a equal minus 1, and b equal y, essentially. Okay, so we will find this. And that is actually turns out to be, okay, 1 minus exponential minus lambda divided by exponential lambda y minus e minus lambda if I define lambda as log of q divided by p. Now I know that q is greater than p, therefore lambda is positive. Okay? So altogether, what we find is that the probability that y is greater than y is of order 1 minus 1 minus 1 minus lambda. Lambda is called a scale parameter, minus lambda y. Here we are. Okay? So we found, in fact, something really important, fundamental. Okay? We found that the probability of high match, probability of a high cumulative score higher than y, okay, is given by a decreasing exponential, okay, with a certain constant here and a certain scale parameter. Okay, so in this model, okay, the probability that we reach a long match, score greater than y, okay, is of the following form. And this is exactly, well, precisely, okay, the same. This is precisely the same as found by Carlin. Okay. Simply the calculation in Carlin is more complicated. Okay. So let me just shortly discuss the scale parameter. Okay. So in the paper of Carlin, analysis of blast. Okay, basically. Of course, we can use this to design the p-values. Okay. And so in the analysis of Carlin, basically, we have this, so same type of equation, okay? But lambda is actually given by a solution. So this is called the scale parameter. And lambda is obtained by solving this equation. Okay, so this equation here is the equivalent of our very simple log QP equal lambda equation, okay, in our model. And as per the C parameter, okay, well, the C parameter is much more complicated. I think I wrote that somewhere, just a sec. I think I wrote that somewhere. So the C parameter, yeah, it's here. Okay. So you can see that this expression is not so super obvious. Okay. And that's the result. This is really a result obtained by Eigelhardt in 1972. Okay. So, but all the fundamental property of Carnot, okay, all the fundamental property of the model of Carnot are basically very much illuminated by this simple analysis. Okay, so also last word. Okay, if you look at the Y max statistics, so what is the probability that Y max is greater than a certain value? Okay, so for this statistic, okay, what is the probability that the maximum excursion Y max is greater than Y? So that's, of course, a complement of the probability that Y max would be smaller than Y. And of course, well, if we assume this is not quite true, but if we assume that these are independent, then it would be 1 minus the product of the probability that the yi, so y excursion I, okay, is less than y, okay. And this would be 1 minus, well, in our model, this is 1 minus 1 minus 1 minus e minus lambda. So this is C actually, and exponential minus lambda y and power m. Okay, where we have m excursions. So this is not totally it is not a complete analysis, but basically if you continue along these lines, okay, you arrive at another kind of distribution, which is called the Gumbel distributions. And the Gumbel distribution is actually a distribution that controls extremal values. Okay, so you basically will arrive at an exponential exp of exp. Okay, exp of exp, and this is actually, you can start to see that here and there, because here Well, it's kind of an exponential here with the power, and here again we have another power. Okay, so the only thing here is that I've not carried the calculation fully because this is not in proper form. But I believe that you can easily convert this formula here to the Gumbel distribution. And this is also a finding of Karner. Okay, I'm going to stop this video here. Okay, so to summarize, well, I have been discussing DNA barcoding. Okay, and I've discussed also, well, I've discussed a bit of sequencing. Okay, how is sequencing done? And also, I've shown you how a simple random walk model of a cumulative score, okay, gets you a long way in the understanding of the probability analysis or probabilistic analysis, statistic analysis of the degrees of matching. I'm Philippe Fussac, and I hope that this video has interested you, and I will stop here. Bye for now.